Welcome to the debate. Today we're diving into really a landmark development in synthetic biology. It's the successful generative design of functional biological systems actually at the whole genome scale. We're talking specifically about using these genome language models, or GLM's EVO-1 and EVO-2, to create, well, completely novel bacteriophages. They've called them EVO-phi. And, this technology is potentially a game changer, especially for creating tailored phage therapies against evolving bacterial resistance.
It absolutely is a technical achievement, no question. Revolutionary, even. But as often happens with these kinds of breakthroughs, particularly in genome engineering, we really need to dig into the fundamental mechanics. What actually made this success possible?
Right. And that brings us to the core question for today. Does this whole generative process, the successful creation of viable phage genomes, does it really demonstrate a fundamental generalizable framework, a kind of new blueprint for getting into novel evolutionary design space? Or is its success still, let's say, tightly confined by very specific template-driven computational constraints? My position is that this really does establish a profound blueprint for designing diverse synthetic living systems, even considering the necessary engineering involved in steering it.
And I'm going to argue that while the results are, frankly, spectacular, they are currently very much contingent upon extensive methodological constraints and a real specificity to the template used. This reliance, I think, limits the immediate generalizability of the framework, especially beyond these very highly characterized small genome templates. What we're seeing is, well, expert engineering, not quite a universal blueprint yet.
Well, I think the evidence for it being a blueprint is pretty overwhelming. This work, it's a huge step forward. We've moved generative design from just single proteins or maybe small genetic circuits to the entire genome level. And the result wasn't just, copying things. It was genuine evolutionary novelty. They generated 16 viable phages. And critically, one of them, EVO-52147, achieved a nucleotide sequence novelty measured by average nucleotide identity, less than 95%. That's basically on par with how we define a whole new bacterial phage species in nature.
Okay, I see the novelty, I acknowledge that, but we have to frame how it was achieved. That success was only possible because the entire process was deeply constrained and just meticulously filtered. The generative ability of EVO-1 and EVO-2, it leaned heavily on supervised fine-tuning, SFT. The models were intensely trained on something like 15,000 existing microviridase sequences. So they weren't just dreaming up possibilities across all of phage space, they were learning the very specific genomic grammar of one very small, very well-understood family.
But they learned those rules well enough to propose functional solutions that, well, they'd never existed naturally before. And it's not just novelty. The generated phages showed superior function. EVO-59, for example, dramatically outcompeted the wild-type template phage in head-to-head competition assays. We're talking a full change in fitness that was vastly higher, 16 to 65 times higher. And another one, EVO-204083, showed significantly faster lysis kinetics. It killed its host almost an hour faster. So the models clearly understood and could manipulate these complex underlying evolutionary constraints to get a real functional advantage.
That's a compelling point about the function, certainly. But have you considered the sheer intensity of the filtering needed after generation to even find those functional ones? I mean, to get those 16 working phages, the researchers had to synthesize and then painstakingly narrow down around 300 candidates. This wasn't trivial. It involved a multi-tiered filtering system, quality control, tropism filters, diversification filters. The success didn't just, pop out of the model fully formed. It was absolutely contingent on this highly specific, intensive computational and biological gauntlet to even ensure viability.
But that filtering, I see it as simply the necessary engineering you need to steer the model effectively. That's precisely what a usable blueprint is. It allows for steerable, user-specified constraints.
I'm not quite convinced it's merely steering when the output seems so sensitive to really minor parameter changes. That suggests, to me, an inherent template dependence.
The whole generation process was guided by this small, historically significant FX174 genome, only about 5.4 kilobases. And the fact that they needed such careful tuning of things like prompt length, just 4 to 9 nucleotides of the consensus start sequence, and the sampling temperature, that parameter controlling randomness, it really shows that success is tethered pretty tightly to staying close to that known template architecture. If this were a truly general blueprint, shouldn't the output be more robust to broader variations in the input scaffolding?
I think that tuning just allows us to precisely target which corners of this vast design space we want to explore. The real significance, I believe, lies in the sophistication of the biological rules that these GLMs manage to capture. Let's go back to the evolutionary novelty point for a moment. The ability to design these functionally complex changes, it really validates the model's grasp of deep biological rules. Take EVO-36. Okay, previous rational engineering attempts had shown that if you just swapped in a G4 phage gene, J, that's a DNA packaging protein, into the wild-type PHX174, the resulting phage was completely non-viable, dead on arrival.
Right. The failure of that rational swap does highlight the complexity involved.
Exactly. But the generative model, working in its context, achieved a novel, compatible structural solution. The EVO-536J protein, the one the model generated, it was significantly smaller than the wild-type J, but it worked. And cryo-EM structural analysis actually confirmed it. It preserved a compatible interaction with the capsid, despite having a very different sequence context. This, to me, proves the generative model captured those intricate protein-protein coevolutionary rules needed for the whole genome to be viable. It navigated constraints that, well, traditional non-AI engineering just couldn't handle.
I agree. The EVO-36 result is quite remarkable. But let's be precise about the novelty here. While it's complex, the design space they explored was still constrained. That G4J protein, yes, it's distant, but it's an element that's known in nature. It's a successful functional reorganization of known parts, not something created purely de novo from scratch. And furthermore, let's not forget the model was deliberately constrained by that tropism filter. It demanded that the generated spike proteins had to maintain, a moderately high sequence identity, at least 60% to the wild-type spike protein.
60% sequence identity still leaves a huge canvas for evolutionary change, especially considering the high coding density in these phages.
Perhaps, but the need for that constraint still points to the fact that functional viability heavily depends on keeping core, known interaction mechanisms intact, like, for instance, host-receptor binding. The necessity of maintaining known tropism points right back to template dependence. And this is where the computational structure itself becomes a key point of debate for me. My concern about generalizability really stems from the very specialized computational tools that were needed to make this model work effectively in this context. Standard bioinformatics tools just weren't up to the task. For example, PHX174's genome is famously compact. It features these complex overlapping open reading frames, ORFs, right? Which makes predicting the genes notoriously tricky. Standard gene annotation tools, they tried 6 different ones. They failed to reliably annotate all 11 genes. So the researchers actually had to build a bespoke, specialized CDS prediction method tailored only to PHX174-like sequences, just to establish the necessary gene-level constraints for the model.
But that level of specialization, I think it reflects the unique complexity of the target organism, PHX174, not some fundamental flaw in the model's core generative power. I actually view the methodology as a strong demonstration of steerable user-specified constraints. That's exactly what you want from a high-utility blueprint. And maybe we should talk a bit about the model internals here, because I think that supports my point. EVO-1 and EVO-2, these weren't just trained on the microviriide. They were pre-trained on a massive data set, over 2 million phage genomes. This huge corpus allowed them to learn the sort of fundamental syntax of viral genomic grammar. And the models themselves use advanced architectures. They incorporate features like specialized attention mechanisms. Sometimes people refer to the underlying math like the fast convolution operations you find in things like hyena layers. These allow the models to efficiently process and learn really complex, long-range dependencies across the entire genome sequence, which is absolutely critical for these compact phages with overlapping genes.
Okay, and how does that specific architectural detail translate into a generalizable blueprint in your view?
Well, it translates into a really strong baseline capability. In the early zero-shot experiments, this is before any of the specialized fine-tuning or filtering, EVO-2 predicted that about 34 to 38% of the sequences it generated were likely viral. And these sequences showed characteristics like high coding density, complex gene arrangements, even high confidence in predicted protein structures, measured by high PLDDT scores. And this was true even when the generated sequences have very low identity to any known natural genomes. So that confirms the models captured these deep features of functional viral architecture before they applied the intensive SFT or built those custom gene-finding tools. The SFT and the bespoke CDS method, that's the rigorous engineering needed to take a powerful foundation model and reliably steer it through evolutionary space toward a guaranteed viable outcome.
It is undoubtedly a profound engineering triumph. But it also highlights the immense complexity of that engineering layer, the human guidance, the bespoke tools required to translate that powerful foundational capability into actual functional life.
Okay, let's pivot then to the immediate implications for therapeutics, because I think this really validates the generative approach as a powerful design strategy. These generative phages showed immediate utility in fighting resistance. A cocktail made from these EVO-phages rapidly overcame PHY-X174 resistance in 3 different E. coli strains. They called them CR1, CR2, CR3, in situations where the original wild-type phage just failed completely.
That is certainly the most exciting potential application. Yes. Undeniably.
And this success wasn't just luck. It came directly from the diversity that was inherent in the generative output. It enabled rapid evolution to overcome the bacteria's defenses. They observed recombination and mutation events involving these EVO-generated phages that led to new high-fitness resistant variants. EVO-FIR1 and EVO-FIR2, where key beneficial mutations popped up on the virion exterior. This, I think, clearly establishes an efficient and potentially generalizable path towards designing genetically diverse, evolutionarily resilient therapeutics, basically, systems designed for evasion.
The potential for resistance evasion is compelling, absolutely. But we need to exercise some caution when we project this success onto future scalability. The tractability of this specific study was almost entirely reliant on that minimalist, historically significant PHY-X174 template. Designing larger phages, let alone much larger organisms, is going to present fundamental challenges that the generative models on their own simply cannot solve.
Are you referring mostly to the DNA synthesis limitations?
Yes, that's a major one. Specifically, cost-effective DNA synthesis and assembly techniques. The difficulty just scales exponentially. And the source paper itself acknowledges this. They state that methodological innovations beyond SFT will be required to handle larger genomes, say even a 50 kilobase phage, which isn't huge by phage standards. We've successfully designed a very small, very minimalist genome where the rules are relatively well-defined and known, but the complexity just explodes for larger systems. It means the specialized filtering, the reliance on bespoke tools that were needed here, well, they would rapidly become prohibitively expensive and time-consuming. And that challenges the idea of a truly universal utility for this blueprint, at least right now.
But that acknowledges the massive challenge of writing the DNA, the synthesis part, not the fundamental ability of the model to design it. The generative model, I argue, has demonstrated the core blueprinting capability. The remaining difficulties are more matters of synthesis, assembly, testing, engineering challenges, essentially, which will inevitably improve over time.
And that, I think, is the crux of our disagreement today. The design works, it's viable, because the overall system is small, and we know its rules intimately. The moment you try to step beyond the Microviridae family, beyond the constraints well defined by the PHYX 174 template, the sheer amount of necessary human intervention, the intensive filtering, the specialized CDS methods needed, it shows that the blueprint, as it stands, is currently contingent on pre-existing engineering tractability, not on some inherent universal generality.
I maintain that what we've seen here is the foundational blueprint. Generative models, especially ones built on these advanced architectural principles like EVO-1 and EVO-2, they can capture the complex, underlying evolutionary design space with enough fidelity to produce novel, functional bacteriophage genomes. This whole sophisticated strategy, it validates the entire generative pipeline as a robust path towards achieving both fitness advantages and, critically, resistance evasion.
And while I agreed it's undeniably powerful, the process, as demonstrated, is currently defined by that intensive computational scaffolding, the highly focused SFT on narrow data sets, and the very specific, necessary filtering against a known small template architecture. It's a spectacular breakthrough in genome engineering, absolutely, but it also emphasizes the significant and highly specialized human guidance still required to safely and effectively harness these generative models at the genome scale, especially if you want to guarantee functionality.
It certainly highlights just how powerful this new technology really is becoming, and perhaps how close we are getting to the deliberate design of life itself.
And it reminds us that the ultimate promise of generative design really depends on whether that necessity for deep human constraint, for intensive filtering, can be significantly reduced as the underlying models and, importantly, the experimental methods continue to improve. Only then might that blueprint truly generalize across the vast diversity of life.